Hello everyone, in this video I'm going to be talking about DNA replication. Now I'm making two videos about this topic, one aimed at GCSE and one aimed at A-level and IB because it is quite a complex process. This is going to be the one aimed at KS4, GCSE, that quite simply explains what the process is and why we do it. But before we come to that process, we need to first of all lay some groundwork. Now, if I draw this, to many people, they'll recognise this as the double strand, or a very simple sketch of a double strand of DNA. And in another video I've spoke about what it's made up of, but we're just going to recap that now. These strands here that form this S shape, they form what's called a double helix, and they are made of sugar, or deoxyribose sugar, and phosphate. And in between those strands, what we have are nitrogenous bases. And you see them sometimes given their letters ATCG, representing the chemicals adenine, thymine, cytosine, and guanine. That's a very basic, simple sketch of a DNA molecule. Now, the reason why I'm showing this now is because I want you to imagine that we unwind this molecule. And if you were to have a hand at the top and a hand at the bottom and they were to twist in opposite directions, what you'd ultimately get is this kind of ladder forming, or it would appear like a ladder. Now, I should say that these bases, each rung on this ladder almost represents two bases that are complementary paired. So A pairs with T, C pairs with G. So, in fact, I'm just going to expand this out a little bit more just to make that a bit clearer. So you can see that each kind of step of two bases paired together. We've got the sugar phosphate sides, or the backbone, holding this DNA molecule in place. Now, a nucleotide, so we need to be clear on this name, a nucleotide essentially is a portion of the sugar with the phosphate attached. So a sugar molecule with a phosphate and an attached base, one attached base, and that we call a nucleotide. So we need to just, and I'm not going into too much detail about nucleotides and polynucleotides in this, but just very basically what a nucleotide is. So it's a portion of the backbone with a base attached, because it's important in this replication process. So let's shrink this and let's think about when we would be doing DNA replication. Well, in another video I've talked about the cell cycle, and that the cell undergoes periods of growth and development, synthesizing all of the organelles. We then have a period of DNA replication, and it replicates prior to mitosis, or cell division. So replication is a part of the cell cycle when the cell is growing, ready to divide. So we replicate the DNA so that we can split the cell and form copies of it essentially. So here's what happens. Let's take our original DNA strand here, and for the sake of argument I'm just going to draw the bases in separate colours here. So this is stage 1. In DNA replication, an enzyme called DNA Helicase unwinds this DNA strand and splits them. So if you imagine it, think of it almost a bit like a pair of scissors here, and it will split them down the middle. So what you end up with ultimately are two strands that look a bit like this, two separated strands. Again, this is sufficient for Key Stage 4 level. I'm going to go in more detail in another video for A level and IB. So we end up with two separate strands. Now the DNA strands are within the nucleus of a cell, and also within that nucleus are what we call free nucleotides. So we have those free nucleotides just floating within the cell. So I'm just going to draw some free nucleotides here, just floating around the cell. Now, I said that A joins with T and C with G via what's called complementary base pairing. And that's exactly what happens. These nucleotides, so if we imagine this one here, if the base on that nucleotide is a T, it would come and find a nucleotide that has a base of an A. So you can imagine if this letter here, this one I'm grabbing is a C, then it would join to where there's a base G. And equally you get the same on the other strand. All these free bases will come and complementary base pair to those two exposed strands. And let's think about what would happen as a result. If you continue doing that, then ultimately you'd end up with two new identical strands forming. So it's only a very rough sketch, but hopefully you can see where I'm going with this. So if you take all those free nucleotides here and complementary base pair them to all the exposed bases on the two strands, we're essentially just recreating our original strand, and we have two, copy one and copy two, two identical strands. We actually call this semiconservative replication. Semiconservative because we're conserving half of our original strand, if you like. So for example, this particular strand that I'm just drawing an arrow to now, that strand there with red bases is actually this original strand here. So in both of our new strands that we're forming, number 1 and 2, we're retaining half of our original. There is another enzyme, and that enzyme is called DNA polymerase, and it's that actually attaches those nucleotides. So students certainly get asked about DNA helicase, the enzyme that unwinds that DNA strand, but here's an extra one to note. DNA polymerase, it's an enzyme that joins those nucleotides onto those separate strands to give what I've drawn in number 3, these two identical copies of the DNA molecule. We actually seal those strands or the nucleotides together on those newly formed strands using another enzyme called DNA ligase. But you certainly need to know about helicase and polymerase primarily. But there is a third one, DNA ligase, which will seal those nucleotides just to strengthen the bonds between them. So this, in a nutshell, is DNA replication. We unwind our original DNA molecule, we split it into two separate strands, we have free nucleotides that via complementary base pairing come and join up, and we create two new strands, both identical copies of one another. Okay, hope all that helps.